Good morning. We are from SGD B2, the first semester of PSSKPD 2021. Today, we would like to present our student project presentation, which title is PCR in Diagnosis of Infectious Diseases. Before we start, I would like to introduce myself. My name is David Christopher Tjandra. My last 3 digits of student number is 066. And now I would like to show our members that will do the presentation. Okay, now we will move on to the first chapter, which is the introduction. COVID-19 has become a health issue of great concern these days. It is a deadly disease caused by the SARS-CoV-2 virus that can be transmitted very quickly through human secretions. The symptoms shown by COVID-19 include high fever, shortness of breath, and dry cough. These symptoms are very common that lead to a confusion for the public in distinguishing whether their symptoms are only a sign of a common cold or cough or a real sign of COVID-19. This confusion was finally overcome by implementing a method of diagnosing COVID-19 known as the PCR test. The PCR, or polymerase chain reaction test, is an examination that is done to diagnose the presence of viruses and bacteria in the human body. In diagnosing specimens, including in the detection of COVID-19, this test has been recognized as the gold standard, as it has a high specificity and sensitivity in diagnosing viruses or bacteria, so that it can produce valid and accurate test results. In the medical world, PCR test is known as an enzymatic method for DNA amplification that happens through an in vitro process. That's all from me. Thank you.
Good morning, my name is Marta, Lawi, and my last 3 digits of student number is 064. Now I will continue David's explanation. So DNA amplification in PCR test can be achieved using implemers, which are known as primary oligonucleotides that consist of 20 until 30 nucleotides. During the PCR test, the DNA fragment obtained from the pathogen in the clinical specimen will multiplicate up to 200,000 times after 20 reaction cycles in 220 minutes. This is also an advantage of PCR compared to other tests. Based on its quality and uniqueness, this PCR test will have the potential to diagnose various diseases if it's carried out in accordance with correct international standards. For example, the diagnosis of infectious diseases can create a new form of education for the public if it's studied thoroughly. As intelligent academicians who think critically and want to dig deeper into the diagnosis of infectious diseases using this test, we have the initiative to identify the reactions, processes, and the types of PCR tests that exist along with their application, effectiveness, and further advantages. This whole discussion will lead us to the essential understanding of the PCR test usage in diagnosing infectious disease. Now let's move on to our problem formulations. First, what is PCR? Second, what are the types of PCR? Third, what is the uniqueness, important steps, and the chemical processes in PCR test, and the last is what types of diseases that use PCR results for diagnosis. That's all from me. Thank you.
I'm Nyoman Mahayana Putra. My last digit number is 050. I will continue about the objective and benefits. Next. Objective. First, to describe PCR. Second, to describe the type of PCR. Third, to describe the uniqueness, important step, and the chemical process of PCR tests. Fourth, to describe the type of disease that use PCR result for diagnosis, especially in the COVID-19 pandemic situation. Next. Benefits. First, for the public community. To educate the public about PCR and its uniqueness, and to increase public knowledge about type of disease that use PCR. Second, for the institution. To adding source of knowledge about the PCR test and as a learning material to introduce students about type of disease that use PCR result, especially in COVID-19 pandemic situation. Third, for the writers, through this assignment, students can expand knowledge about PCR. Students will also know about the basic principle and overview of PCR, and students can also find out the types of disease that use PCR result to diagnose disease. That's all for me. Thank you.
Good morning, my name is Fernando Ghosali, and my last 3 digits of student number is 036. Now I'll explain about the definition of PCR test. Next. Polymerase chain reaction, or PCR, is a revolutionary biotechnology method to multiply DNA sequence. PCR was developed by Carl Mullis in 1980s. This technology actually has great effects in scientific research, such as the Human Genome Project. PCR is also used for clinical diagnosis, especially to detect pathogens. Main components that are needed in PCR include DNA template, DNA polymerase, primers, and nucleotides. DNA template contains the specific sequence that extracted from the subject. Primers help DNA polymerase by providing a starting point. PCR involves three processes. They are denaturation, annealing, and elongation. PCR can multiply DNA fragments to millions or billions of copies. Despite its sensitivity, any contamination of the test subject can lead to incorrect results. That's all from me. Thank you.
Morning, my name is Edgar, and my last three digit of student number is 045. Next. PCR test process require the mixing of DNA template, a pair of oligonucleotide primers, thermostable DNA polymerase, TNTPs, and buffers, mixed in a test tube or 96 well plate. The process of PCR consists of three main steps. First, the reaction mixture is heated to more than 90 degrees to allow the separation of the DNA double helix into single strand. This process is called denaturation. Then the temperature is lowered to 5 degrees below the melting temperatures of the primers. These steps allow the primers to attach to its opposite end of the target DNA and complement each other. This whole process is known as annealing. Unless the temperature is raised once again and the DNA polymerase is starting to extend start from the primer side by the nucleotides to form a new DNA strand. This last step is usually known as extension. The repetition of the PCR steps doubled the number of copied DNA exponentially. After the product is obtained, the next thing to do is to visualize the product. That's all from me. Thank you.
Good morning, my name is Gusti Agung Saraswati. My last three digit of student number is 056. Now I will explain about the PCR result. The PCR result can be affected by many variables. One of them is titration, concentration of magnesium chloride in DNA polymerase. Component required to prepare PCR, number one, thermostable DNA polymerase to catalyze template-dependent DNA synthesis. Number two, a pair of synthetic oligonucleotides acting until 28 nucleotide base long as primer in DNA synthesis. Number three, accumular amount of DITP, DTTP, DCTP, and DGTP are found in standard PCR. Number five, divide contents. All thermostable DNA polymerase require free defined contents for the activity. 5, buffer to maintain pH between 8.3 and 8.8 at room temperature. 6, maneuver contains. 7, template DNA templates containing target sequence can be added to the PCR single and double standard form. That's all for me. Thank you.
I'm Nevinne Nathania Susanto. My last three digits of student number is 051. Now I'll be explaining about the types of PCR tests. The basic methods and principles of PCR have been developed into various types. Each variant has its own advantages, disadvantages, and specific use. The most commonly used types of PCR are, firstly, there is hot start PCR. Hot start PCR is used to increase optimal production of the amplified product that is needed. However, its disadvantages is that the whole process must be done manually and the temperature must always be kept high. There's also a risk of high contamination happening during the process of adding the final reagent to the mixture. Secondly, there is chemical hot start PCR. It modifies co-solvents and enzymes in the process of primer reduction. Some specific co-solvents, such as dimethyl sulfoxide and formamide, are able to increase the precision of the entire PCR process. Enzymes such as uracil and glycosylase are also commonly used in this method. Third, there is touchdown PCR. This method of PCR is used when the specificity of the PCR needs to be increased and the off-target priming needs to be decreased. The process of touchdown PCR involves an assortment of annealing temperatures. That is all from me. Thank you.
My name is Janice Wijaya, and my last three digits of student number is 037. Now let me continue Nevinne's explanation. So the next type of PCR is degenerate PCR. This type of PCR involves intentionally decreasing the specificity of the process so the detection of divergent sequences can occur. Degenerate PCR has been known to successfully detect and diagnose several norovirus pathogens. The next one, we have nested and hemi-nested PCR. Yes, thank you. These variations of PCR are mainly used to increase PCR sensitivity. This is done by increasing a primary PCR product with the presence of the secondary PCR. The difference between nested and hemi-nested PCR lies in the second round of PCR. Next, we have multiplex of PCR. This PCR variation involves the simultaneous amplification of 2 or more DNA sequences. This process requires similar annealing temperatures so the amplification results can be equal. Failure to do this will result in dimerization, lower sensitivity, and competitive inhibition. Next we have reverse transcription PCR. Reverse transcription PCR amplifies RNA targets and couples it with a reverse transcription reaction. This variation of PCR is commonly used for gene expression detection and for diagnosis of infectious and genetic diseases. The last one is the quantitative PCR. This method of PCR measures how much the target DNA or RNA is in a sample. It is also called real-time PCR or qPCR. Real-time PCR is most commonly used in analyzing gene expression and also for pathogen and mutation detection. That's all for me. Thank you.
Morning, I'm Komang Ayu Widhiani. My last 3 digits of student number is 044, and now I'll explain about the uniqueness of PCR test. PCR is hardly regarded as an accurate method to detect viruses because it is a simple and easy technology to implement. It also boasts other advantages such as being highly sensitive, rapid, and also accurate. As it is shown in study by Igloisi et al. Showed that PCR has an efficiency rate of 90%. Besides that, PCR consumes less time and also more cost-effective when compared to other similar technologies. PCR is even nicknamed as the gold standard. Some reports also indicating that RT-qPCR has the ability to target bacterial pathogens. Next slide. I'll continue to the next discussion about kind of diseases that use PCR result for diagnosis. The first is tuberculosis or TB. It is an infectious disease that attacks the lungs caused by Mycobacterium tuberculosis. For diagnosis and therapy, early detection of MTB is needed. Using multiplex PCR can facilitate the difference in MTB speciation. The second one is hepatitis B. HBV can cause these infectious diseases. HBV can be detected using PCR. ddPCR can detect HBV sensitively and specifically. And the third one is streptococcal pharyngitis. Streptococcal pharyngitis can be diagnosed using PCR. And empirical treatments such as the use of antibiotics and therapeutic cause of pharyngitis can be limited by the use of PCR. That's all from me. Thank you.
I'm Ledwin Meikel Wibisono. My last 3 digits of student number is 061. Now I'm going to continue my friend's explanation about kind of diseases that use PCR result for diagnosis. Atypical pneumonia. Atypical pneumonia is mainly caused by Chlamydia pneumoniae, Mycoplasma pneumoniae, and Legionella. This disease can be diagnosed using multiplex PCR. The next one is Lyme disease. Lyme disease is caused by the bacterium Borrelia burgdorferi. To avoid misdiagnosis, the PCR test is one way to detect DNA from Borrelia burgdorferi. The next one is human immunodeficiency virus, or HIV. In the primary stage, HIV do not have specific symptoms. Therefore, HIV infection is difficult to get an early diagnosis of, resulting in opportunistic infections. In diagnosis, PCR tests are used to detect HIV. The next one is acute febrile illness. Acute febrile illness can be identified using PCR. For example, malaria falciparum. The PCR used to detect this illness is a single PCR. And the last one is SARS-CoV and COVID-19. The SARS-CoV and COVID-19 are diseases caused by the coronavirus. In diagnosis of COVID-19, the real-time reverse transcriptase polymerase chain reactions, or RRT-PCR technique is used. That's all from me. Thank you.
Morning, my name is Nyoman Ayu Medhira. My last 3 digits of student number is 070. Now I will explain about application of the PCR test in infection disease. Because of the PCR-based test development, several variations of the PCR-based technique emerge with their own specification in identifying infectious disease. First is multiplex PCR or MPCR. With MPCR technique, it is possible to detect several pathogens at the same time with just one specimen. This technique is used in detection of sexually transmitted infections like gonorrhea, chlamydia, syphilis, and even HIV. Second is loop-mediated isothermal amplification or LAMP-PCR. With this type of PCR-based test, the final product that was produced has almost reached 109 copies of target DNA in only less than an hour. Third is real-time PCR or qPCR. qPCR is a clinical diagnostic that uses quantitative PCR as its method for detecting the presence of infectious agents. That's all from me. Thank you.
I'm Amanda Devasya. My last 3 digits of student number is 068. Now I will continue the presentation. So next is dPCR or digital PCR. dPCR can detect virus even in a very low viral load and low concentration of sample. dPCR could be used in the diagnosis of many infectious diseases caused by bacteria to parasites such as HPV, human influenza, HIV, Plasmodium vivax, and etc. Next, the reverse transcriptase PCR or RT-PCR. Nowadays, RT-PCR is mostly known as the test for COVID-19. The sample of RT-PCR assay in detecting COVID-19 is collected from the upper respiratory system. Next, I will explain about effectiveness of the PCR test in infectious disease. Due to advances in medical science and molecular biology, in general, the diagnosis of complicated disease uses molecular techniques like PCR, different blood, and genetic diagnosis as a choice to have first diagnostic modality. PCR is the most commonly used diagnostic modality. From the result of the literature study, the specificity of the rapid test antibody range from 91 until 100%. The RT-PCR method has the advantage of being able to detect antigen at low concentration, but RT-PCR has drawbacks such as, first, requiring expensive equipment and examination costs, second, requiring laboratory personnel with special skill, third, long processing time around 2 until 3 hours, and last, high risk of exposure. Thank you, that's all for me.
My name is Yovilia Tio Yunus. My last 3 digits of student number is 035. Now I will explain about conclusion and recommendation. Next, from the study above, we can conclude polymerase chain reaction, or also known as PCR, is a revolutionary biotechnology method to multiply DNA sequence. PCR has been developed into various types, such as hot star PCR, chemical hot star PCR, touchdown PCR, and etc. PCR is often nicknamed as the gold standard in several clinical diagnoses because of its ability to recognize various kinds of pathogens rapidly and accurately. In the PCR test process, the components such as template DNA, nucleotides, primers, and DNA polymerase will be mixed together in a test tube, then placed in the thermal cycler machine. PCR is used to diagnose several diseases that is caused from various pathogens like bacterium, fungus, and viruses. Next, I will explain about the recommendation. The main and crucial thing is for this topic to be further studied by scientists or researchers, especially in the biomolecular field, pharmaceutical, and medical students. By understanding the important steps in the PCR chemical test process, more knowledge can be obtained regarding how it is supposed to work. That's all for me. Thank you.
That's all for our student project presentation today from SGD B2. Thank you for listening and giving your attention. We are very sorry if there are some mistakes during our presentation, and we hope that our student project can inspire and motivate lots of people based on its benefits. Lastly, I would like to close it with the Paramasanti Om Santi 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 Om. Thank you very much.